moving on into video 4. Video 4 and 5, these two are going to be interchangeable because they're kind of sidestepping the main idea of everything that we've talked about and giving a little bit more kind of specific examples and diving a little bit deeper into stuff that we've already talked about. So this first one, we're going to talk about the contrast and differentiation of our gated channels. So looking at ion channels, we have two different types. Remember, so when we talk about facilitated diffusion, we had the ion channels and the carrier proteins. We're going to focus in right here on the ion channels and what they do and kind of what those look like. So we have ungated channels, where this is a channel that's ungated, it's unblocked, it's always open, letting things pass through whenever they want to, okay? With these ungated channels, the transport or the crossing of material is determined by the size, the shape, and charge of both the channel and the ion. So these channels are still specific for certain things to allow them to come through. Okay, not just anything can pass through willy-nilly. It still has the specifications to let specific things come through. One of the examples of this is this thing called leaky ion channels, also known as funny channels when you talk about them in the heart. Leaky ion channels do slowly let certain things, mostly sodium. We have leaky sodium channels. Like stuff slowly leak and trickle through, and they're open all of the time. Okay. The main one that we're going to focus on are our gated channels. I'm going to add one here as well. There's also mechanoreceptor channels. Main two we're going to focus on are our voltage gated and our chemical or ligand gated. Okay. Voltage, we'll start with voltage. Voltage gated ion channels. There's two big ones that we're going to talk about for right now. We'll add more in the future. We're going to talk about voltage gated sodium channels and voltage gated potassium channels. Okay. This is going to skip ahead a little bit. And we're going to come back to a lot of this stuff. So right here, this is going to be kind of the last video of this series. This is called an action potential. What this graph shows, this is showing the membrane potential at any given time, specifically of a nerve cell as it goes through sending a message. Okay? What I want to focus on here is we have what's called a resting membrane potential, where the electrical difference between inside and outside is when the cell is at rest, and that's maintained. It gets maintained for a nerve cell around -70, and we'll look at exactly how that is maintained in the next video. We can have stimulus that's going to cause change in this right here, in this in the membrane potential. Certain actions, movements, stimulations, that kind of stuff is going to cause what's known as depolarization, which is things going from negative to positive. And along this way, as we go up, when we reach certain voltages, voltage-gated channels are going to either open or close. That's why they're called voltage-gated. When we get a change in the membrane potential, or in the voltage of our cell, we get gates that will open and will close. Now, big ones right here that we're going to talk about right in this first week is going to be the sodium and the potassium. We might also touch on the calcium ones as well, if we can get to those. I'm just going to touch on this. We'll look at this a little bit more later, but this is the sodium gated channel and the potassium gated channel shown blown up a little bit more. Okay? So sodium channels are different because they have two gates. Okay, so we call these either gated channels. We have an inactivation gate and an activation gate. Okay? The way that this is going to work, when the activation gate opens, the channel is activated and sodium can go through. Once the inactivation gate closes, it shuts off the channel, and it's no longer active. And then at a different point, these are going to reset back to their original position. We'll get more into these when we talk about action potentials. The potassium gated channel just has one single gate. You can call this an activation gate, but you don't really need to because there's not a need to distinguish them from another one. So the potassium voltage gated channels is either open or it's closed. It's either active or it is inactive. Okay, and we'll come back to this a little bit later on. Okay, so that is our first one that we wanted to look at. Our voltage gated. We're also going to have chemical gated ion channels. This we're going to look at kind of in the next chunk of material that's for this week when we start looking at the neuromuscular junction. We have examples of we've seen examples of these kind of these chemically gated processes or something similar in Biochem 1. If you remember the G protein coupled receptors, that's not exactly the same thing, but it's similar. We have a chemical ligand that's going to come and bind, and in G protein coupled receptors, we had the whole cascade with cyclic AMP and all of that good stuff. This is much, much simpler. The ligand or the chemical is going to come in, it's going to bind to the ion channel, and the actual physical binding of that ligand, aka that chemical, is going to cause the gate to open and allow stuff to cross through. Most common one that we're going to see are these nicotinic acetylcholine receptor channels. Okay, these are the big ones. You've heard the word nicotinic, you've heard the word muscarinic. We'll touch on those a little bit in the future as well. And this is something that's not going to go away. You're going to see these kind of words, these nicotinic acetylcholinergic or muscarinic channels as well. You're going to see these pretty often throughout the program. There's a lot to do with like pathology, medications, and general physiology that do touch on these kind of things. So start putting these into that file of I need to remember this. Okay, so that is the different types of gated channels. The professor might touch more on mechanical receptors. They're not super complex. Biggest thing is like they have to do a lot with mechanical receptors. They have a lot to do with touch, like when you touch something and press something. The actual like pressure on you is going to cause, especially like on nerve cells, like that pressure is going to open mechanoreceptor channels to allow ions and stuff to flow, to generate like action potentials and like the actual sensation. Action potentials are the messages travel along nerves to activate the nerves to send messages. So those are what those mechanoreceptors. If your professor goes more into those, I'm sure you can look up more information specifically about that. Okay, so that's this video for our gated channels. In the next one, we're going to talk about the sodium-potassium pump. And again, just like I said, this video and the next video, you can really kind of interchange between the two of them. Hopefully, we'll be able to get this done in just one or two more videos. The last video is probably going to be a little bit longer just because action potentials have a lot more information within them. Thanks for watching, and we'll see you on the next one.